In this section, first, I'd like to use a couple published studies to show how to model neurodegenerative diseases with PSA-derived neural cells. Then I will introduce our STEMdiff 2D neural products, including forebrain neuron, midbrain neuron, astrocyte, and microglia kits. I will also show some in vitro co-culture application using our products at the end. As Irene mentioned in the previous section, a lot of studies have been done with animal models. However, studies of transgenic animals and neuronal cells overexpressing disease-related mutant proteins may lead to mechanisms which are different from those in patients. It's important to combine patient cell-derived hPSC models with animal work to increase the human relevance. As we can see in this figure, researchers can generate various types of neuron and glia from patient cells. Then they can use these cells to study the mechanisms of these diseases or carry out drug tests of neurodegenerative diseases, such as AD, PD, ALS, and many other diseases. For example, in this study, researchers did a drug screen with familial AD hPSC-derived neurons and found cholesterol metabolism can regulate tau and A-beta. They also investigated the mechanisms using these neurons. Here are some data of this study. Left panel showed effects of different statins on phosphorylated tau, total tau ratio in hPSC-derived neurons. We can see the dose-dependent effect from this statistic data. Interestingly, they also generated astrocyte from iPS cells and found some statins may reduce the viability of astrocytes at high concentration. The authors of this study took advantage of iPSC-derived neural cells and showed us a potential druggable target. Here is another example of applying iPSC-derived cells to the research of AD. First, the researchers used CRISPR-Cas9 to generate ApoE4 iPSC cells from ApoE3 iPS cells. Then they differentiated these cells to neuron, astrocyte, and microglia-like cells. RNA sequencing data showed differential expressed genes between ApoE4 cells and ApoE3 cells. Also, they found a significant number of neuronal as well as microglia signature genes exhibited concordant ApoE genotype-dependent changes between the human samples and their iPSC-derived cells. Furthermore, they showed that expression of these ApoE4 associated genes in each cell type exhibited significant correlation with clinically defined AD phenotypes in human patients. Now let's look at the phenotypes that they found in each cell type. Here is the data of ApoE4 neurons. RNA sequencing data revealed over 400 differential expressed genes between ApoE3 and ApoE4 neurons. We can see here cell proliferation-related genes were downregulated by ApoE4, while neurodifferentiation-associated genes were upregulated. Figure C to figure F showed increased number of synapses, early endosomes, and A-beta secretion in ApoE4 neurons. So how about astrocyte? First, they showed that the ApoE protein levels were reduced in ApoE4 astrocytes. They also found about 1,300 differential expressed genes between ApoE4 and ApoE3 astrocytes. Then they measured the cellular level of cholesterol in astrocytes and found stronger signals in ApoE4 versus ApoE3 astrocytes. Interestingly, their data here indicated that less A-beta was taken up by ApoE4 astrocytes. Next, they investigated microglia-like cells. Again, RNA-seq data showed over 1,400 differential expressed genes, with a third of upregulated genes in ApoE4 microglia-like cells were associated with immune responses. Figure C indicated ApoE4 microglia-like cells took up A-beta much more slower than ApoE3 cells. Then the authors took one step further by using a co-culture system of microglia-like cells with mutant cerebral organoids that display A-beta aggregates. As we can see from figure D, A-beta accumulation in organoids containing ApoE3 microglia-like cells was significantly less than the single culture group. However, the mutant organoids co-cultured with microglia cells carrying the ApoE4 cells exhibited more extracellular A-beta aggregates than those co-cultured with ApoE3 cells. These two studies we just discussed clearly show that the disease models based on hPSC-derived neural cells are powerful tools which can help researchers understand the mechanisms of these diseases. So, if you want to generate hPSC-derived neural cells, we are here to help. Actually, in STEMCELL Technologies, we have both 2D and 3D differentiation products. Because we only have limited time today, I will focus on our STEMdiff 2D neural-related products. I will introduce 4 products today, including forebrain, midbrain, astrocyte, and microglia kits. Here is the diagram of our products. You can use mTESR1 or our new mTESR Plus to culture PSC, and use STEMdiff neural induction kits to generate neural progenitor cells. Then you can either maintain them in our neural progenitor medium or bank the cells for future use. Or you can use our 2D neural differentiation kits, which I will talk about in detail later, to make neurons or astrocytes. If you want to produce microglia, we provide hematopoietic kit to differentiate PSA to hematopoietic progenitor cells, which can be further differentiated to microglia by our microglia kit. With these products, you can model diseases that you are interested in and perform a series of downstream applications. Okay, first, this is our STEMdiff forebrain neural system. As I mentioned before, this system includes neural induction kit, forebrain neuron differentiation, and maturation kits. It's really easy to prepare the medium as each kit only has one basal medium and one supplement. To generate forebrain type neurons, you can start from either EB protocol or monolayer protocol. The process is very straightforward as shown here in this diagram. We also provide algorithm for generating EB, and STEMdiff Neural Rosette Selection reagent for rosette selection. After 18 to 19 days differentiation, you can culture your neurons in our brain phase-based maturation medium for either long-term or short-term application. This is the monolayer protocol. Although it will take longer time to get neurons, it's also simple to work with. After 2 weeks in maturation medium, you can expect over 90% T-UGL1 positive neurons and less than 10% G5 positive astrocytes in your culture. And the majority of these neurons are forebrain type neurons, indicated by this FOXG1 staining. Here are the STEMdiff midbrain neuron system. It has a quite similar format as forebrain neuron system. 3 kits, 1 basal, 1 subelement each. Just like the forebrain neuron system, you can also start from EB protocol or monolayer protocol for our midbrain neuron system. The protocol is slightly different from forebrain kits, but it's also very straightforward and easy to work with. This is the monolayer protocol of midbrain kits. With this product, overall, you can expect over 15% TH positive dopaminergic neurons with over 90% T-UGL1 positive neurons, and less than 10% G5 positive astrocytes in your culture. And immunostaining data showed that these midbrain type neurons express dopamine active transporter. Okay, let's talk about our STEMdiff astrocyte system. This system also has similar format, besides there are 2 supplements in the maturation medium. And like the forebrain and midbrain system, astrocyte can be generated via either EB protocol or monolayer protocol. Although the whole process will take almost 8 weeks, the protocol is not difficult to handle. Meanwhile, there are 2 freeze points in this process, one at the neuroprogenitor cell stage, one at the astrocyte precursor stage. You can freeze down the cells at these 2 time points and store them for the future usage. Again, the monolayer protocol of astrocyte kit will take longer time in the neural induction process. As we can see from these images, our products can help you to get over 70% S100B positive and more than 60% G5 positive astrocytes with less than 15% double coding positive neurons. Our beta tester used our astrocyte kits, and their data showed that besides S100B and G5, our astrocyte express a series of astrocyte markers, as shown here in the figures. We also carry out calcium image assay with ATP treatment of astrocyte. This is a well-established assay to show astrocyte function. Here is the representative images of calcium image assay. After we added ATP into the well, these astrocytes showed a robust increase of calcium signal. Statistical data also showed that PSC-derived astrocytes had a significant higher increase of calcium signal in compared to control groups. The last product that I want to introduce today is our STEMdiff microglia system. This system is quite different from previous ones. It has 3 kits with basal medium and multiple supplements. More importantly, this protocol is different because it has 2 steps. First, we need to generate hematopoietic progenitor cells with our hematopoietic kits. To start with, high quality PSA and desired colony density is the key to success. And this step will take about 12 days. After that, we culture these progenitor cells with STEMdiff microglia differentiation medium. 24 days later, medium will be switched to STEMdiff microglia maturation medium. We know having a high quality hematopoietic progenitor cells population is critical for later microglia differentiation. Luckily, our hematopoietic kit can generate more than 95% CD43 positive progenitor cells. Our data showed that STEMdiff microglia kits can consistently produce over 80% CD45, CD11b positive, and high percentage of TREMP2 positive cells. We also tested microglia-specific gene expression in our cells. Compared to circulating monocytes, our cells had an increased expression of these 7 markers as shown here. Now, next question would be whether our PSA-derived microglia is functional. We can see from here, from these images, at day 34, our microglia was able to take up pH-sensitive bioparticles, as well as fluorescence-labeled A-beta. As we know, not only neurons, but also other types of cells in the nervous system play critical roles in the pathology of neurodegenerative diseases. Therefore, single culture of certain type of neural cells may not be able to recapture the disease pathology accurately. In order to have a more physiological model and to study the interaction between neurons and glia in the context of diseases, researchers have developed co-culture system to mix different types of cells together. Today, I'd like to share with you some co-culture application using our STEMdiff products. This is neuron astrocyte co-culture. Our data showed that forebrain neurons and astrocytes produced by our kits can be cultured together with our forebrain maturation medium. Interestingly, neurons with astrocytes had longer neural right outgrowths, as well as better survival rate compared to control group. We also checked the synaptogenesis in the co-culture system. As shown here, neurons in the co-culture group seems to have more puncture-like synapsing signal. Our preliminary tests showed that our forebrain neurons and microglia can be co-cultured together with modified brain-phase neural medium. Here we used EBA1 to stain microglia and TUJ1 for the neurons. We also try to mix our forebrain neuron, astrocyte, and microglia together. Basically, we seed our astrocytes on top of forebrain neurons. Then after 1 day, we seed microglia into the cell. Our data here show that these 3 types of cells can live in the same well. As I mentioned in the very beginning, besides 2D neural products, STEMCELL technology also has 3D neural products. In this experiment, our microglia were added to dorsal forebrain pattern brain organoid produced using STEMdiff dorsal forebrain organoid kit. We can find microglia indicated by EBA1 staining in the organoid. Interestingly, if we introduce an injury on the co-culture by piercing the organoid with a needle, then we can find the microglia displayed an activated amoeboid morphology as shown by EBA1 staining here. These 2D and 3D co-culture systems set up by our STEMdiff products are powerful tools for disease research. To summarize today's presentation, we know the neurodegenerative diseases can't be fully captured with just one system. So combination of hPSC-derived models with animal work can increase robustness and human relevance. With STEMCELL technology provide teaser media to support high-quality human PSC culture. And our brain-phase neural medium can increase physiological relevance and better support neural function in vitro. Moreover, STEMCELL technology provides STEMdiff 2D and 3D PSC-derived culture system to study the mechanisms of diseases. If you want to know more about neuroscience research or neurocell culture, you can use these links to access the related resources provided by STEMCELL technology. That will be all for today's presentation. Thank you for joining us.